So I think that without hesitation, we can start. So basically, I would like to start with a question that I would like to ask to you, and is, can we bring resources from Earth to other planetary bodies? But the answer, obviously, is yes, we can bring resources from Earth to other planetary bodies, but the problem is that could be environmentally and economically not suitable, because we need to destroy our planet to colonize or to be settled on another one. But there's something that can help us, and in this case, could be in situ resource utilization. I don't know if some of you already know about this technology, but basically, as the name suggests, is the utilization of resources directly on other planetary surfaces, such as can be moon, can be asteroid, can be comets, maybe in the future, can be whatever you want in space. You need only to have a surface. So at some point, we can extract resources directly in situ in space. But in situ resource utilization can be splitted in two different parts. One that can be considered abiotic, and the second one that can be considered biotic. The abiotic one is without the utilization of living organisms, while the biotic one is the one in which we can use plants or microbes or bacteria or whatever we want to extract the resources directly in situ. And today we will focus only on the metal extraction through microbes. But we don't have to think to something like this that environmentally it's really ugly, in my opinion. So we don't need to think about open pit mine or big mines on Earth, but we need to think something more like this one, in which we have ores and waste coming from other kind of mining operation, in which we have on the top two simple stuff. The first one is an acidic solution in some cases, and the second one is a microbial community or a single microbe. At some point, you have only to wait for days, and the microbes naturally, through their metabolism, are able to extract metals from the minerals into the ores and bring them in solution. At some point, when they are in solution, we can apply electrochemical techniques to be able to recover all the metals in which we are interested. And you have to think something that is important, in my opinion, is that the 30% of the global copper utilized today for your phones or for your PC or whatever you want come from biomining, from the utilization of microbes for extraction of metal. So 30% of copper is a quite big number. Also, the 5% of gold is extracted through this method. But to understand how to perform this kind of experiment, I need to move to Scotland in this wonderful place that is the Highlands, because there's a group that already tested this kind of techniques in space on the International Space Station. And the group was at the UK Center for Astrobiology, so I moved there and I started my experiment. So we understand that we need only two stuff, a microbe and a substrate. First of all, we choose the microbe. The microbe that we used was Sphingomonas desiccabilis. That is not an extremophile, but it can be considered like one of them because it's able to survive in many different environments. And as I show you now, it's able also to extract some metals. So we choose Sphingomonas desiccabilis because also the name suggests is desiccabilis can be bring in space, because to bring microbes in space, we need to be desiccated. And the second stuff was to choose the substrate. So we moved to Canada and we chose to take 5 different samples of Canadian mines known to be full of platinum group elements and rare earth elements, and also they can simulate the impact crater on Mars, for example. The second kind of substrate that we took was an Icelandic basalt that we can use to simulate the regolith of the moon, or maybe the regolith of Mars, as you want. The last samples that we took come from space. Is an extraterrestrial sample. It was one of the meteorite coming from Vesta. Vesta is a nanoplanet in the asteroid belt. So we took a piece of this meteorite, we smashed it, and what we did, basically the experiment is pretty easy. You need to take the powder, the samples, the substrate, and add the microbe inside this medium that could be M9 or whatever you want. And at some point you need to wait. After you wait, you can start making your analysis. And the first thing that we noticed, the first thing that we analyzed was the mineralogical composition of the substrate through the XRD, X-ray analysis. And we discovered that all the samples coming from the mines are basically massive sulfide deposits. So basically are full of sulfides, and sulfides are really able to connect with metals. So at this point, we have a lot of metals. And they are similar, as I already saw before, are similar to impact crater on Mars. The last 2 samples, the basalt and the eucrite, are more or less similar in composition and are full of plagioclase that can be utilized as simulant of lunar regolith or Mars regolith. Then what we saw that Sphingomonas desiccabilis was pretty able to survive and to grow on average sample sort that we used, because we saw the massive production of biofilm. On the left, you can see the biofilm that is colored through a dyer, and the green part is biofilm, while the black part is the rock. And on the right part, you can see an SEM picture in which we can see really a lot of microbes connected to APS and to biofilm. Then what we did? Basically, we did an optical density analysis to understand if we have microbes in our samples, and we discovered that in all the samples that we analyzed, also in some of them there was present arsenic and lead, we found that the microbes were present in a less quantity than in the negative control because of the composition of the mineral, but we saw that it was able to grow in every single samples. Then we asked to ourselves, but are alive or not these microbes that we can see through the optical density? So we performed colony forming units, and we discovered that in all of the samples, microbes were alive, and they were actively eating the substrate, the rocks. That is amazing in my opinion. And then we check which is the yield of extraction of the elements that we are interested into through ICP-MS analysis, and we discovered that Sphingomonas desiccabilis is pretty able to mobilize a lot of different elements from the periodic table. We can build a periodic table specifically for Sphingomonas, and it's another stuff that, for my opinion, is really interesting. And then we can focus only on single results because all of them are more or less similar. And we discovered that Sphingomonas desiccabilis is able to extract a lot of different elements between transition metals, tungsten, iron, manganese, and whatever, but is also able to extract rare earth elements like platinum, palladium, and germanium, and all of these elements can be used in metal sintering. Lastly, is also able to mobilize uranium that is interesting not only from a biomining point of view, but also for bioremediation and biorecovery. So at this point, we have this microbe that can be used for 3 different technologies. So what can we say? We can say that utilizing Sphingomonas desiccabilis for this kind of experiment has some advantages, like the natural pH, because usually these kind of microbes lives in acidophilic environments, in acidic environment, and are considered acidophiles. But in this case, we can avoid the problem of acid mine drainage and these other kind of environmental problem. And also, we can say that this kind of microbe can be sustainable for future space exploration and long-term exploration in space and long-term settlement. And then at the end, we can say that this kind of method that we can perform in small vials can be can be done on a big scale. And that's all. That's me. I finished. Thank you to everyone.